So next up, we've got Dave Green. It's going to be talking about CCP, ARIES and the genomics facility that we've got.
Right. Oh, good morning. Thank you for persevering with us this morning. So this is the third sort of element to the CCP-ARIES Centre. I think in your handouts it might say genetics, genomics. It's kind of it's a mishmash of words. It's a little bit semantic in terms of the issue. But what I wanted to introduce really is not so much the research that we're doing, but actually just introduce what we can do or some of the potential that we can offer to projects. So as Mikey has outlined, this is a facility, and while we don't use the term one-stop shop, but it is in some senses built and constructed in that way to allow us to come here to a remote location, and you can stay here and do pretty much everything or a lot of things that you need to for research projects. And so that, and at the core of that is that algal cultivation and the expertise. And I think that's really our strength, is that we have a very diverse culture collection, and we are culturing these organisms all the time, and we're hitting problems left, right, and center, and we become very adept at solving problems. So while we're not experts in genomics, in metabolomics, maybe in structural chemistry, we can do the biology very, very well. And I think some of the questions that you want to answer, that's what you need to get that right, and then everything else will flow from there. So it's one-stop shop, but it's also there are independent elements to this facility, and you can access any one of these things without having to interact with others. So in this slide, what I'm just going to outline is some of the things that we can definitely do, and we do very well, and we do routinely. So this is our current capacity. In terms of the UKRI's NERC's contribution to the facilities in CCAP, we were able to get a little bit of extra instrumentation just to sort of take away the tedium of DNA extractions. And also, more specifically for the genomic side of things, we've got an agarose gel-based system that allows us to size select and also purify high molecular weight DNA, which is a critical component of genomics research. Again, it's a sort of one-shop sort of concept. We really want to be able to do as much as possible in one place without having to sort of transport between here and Glasgow, because that's an issue. So this is how we've kind of structured what we do and what we can offer. So the first thing is, certainly when I was taught at school how to use a digital calculator, we were told that rubbish in equals rubbish out. Any genetics experiment, if the quality of your DNA is not up to the job, you're going to have a nightmare. And also it will affect your results hugely downstream. So the quality of your DNA that you get out is a prerequisite for good research subsequently. And the data I'm showing here is work by Frederick de Boer, who will speak a little bit later. This is work he did with seaweeds, and they represent one of the most challenging algae out there in terms of getting high quality DNA. And actually this project was also linked up to a collaboration with colleagues in NYU who are actually genome sequencing, and we are now assembling the metagenomes from that in conjunction with these colleagues. But so Fred has gone back into the literature and found some old tricks, and we have substantially been able to improve the methodology for getting high quality DNA out of seaweed. And we can also use these techniques and tricks to post-process a DNA that's not good enough quality. Again, featuring Frederick's work, this is from his PhD, and we have some expertise and skills in getting high quality RNA out of a range of algae. So in this case, it's Nanocoropsis. If you look at Fred's forearms, you'll see that they're very strong. That's because each sample took about 15 minutes of grinding liquid nitrogen. But but he did get extremely good quality RNA, and this experiment is where he's actually analyzing the gene expression of algae and bacteria at the same time. And we were asking the question, we've got a bacteria that likes to kill the algae, we've got a bacteria that likes to protect or block that killing activity, and we want to understand how all three organisms are interacting at the same time. So that's what this work was about. We also have done work with seaweeds in terms of getting RNA. So we have a PhD student writing up at the moment who has, one of her chapters is looking at gene expression associated with the sugar kelp, Saccharina vittissima. There's no pretty picture here. Polymerase chain reaction is a very common technique, as with DNA extraction. These are things that we do all the time. And most of the time it's very trivial and it works beautifully. But not when you start swapping between different protist groups and algal groups, it can become quite a nightmare, such that it can actually stall and be quite a major impediment. So colleagues here at SAMS were working with the Global Seaweed Star Project, and we're receiving hundreds of samples of red seaweed from all around the globe, and they're trying to barcode those organisms. There were hundreds of DNA extractions and PCRs. They pretty much all failed. Fred's techniques were then applied to post-process that DNA, and then pretty much that work has now gone ahead, and actually we've got some nice mitochondrial DNA sequences out of it, I think. People call them contaminants. I'm actually a bacteriologist, so I call them friends, and some of them are foes, but in algal cultures, algae and protists don't typically live by themselves, both in the culture, but also in the environment. So I'm very interested in the microbial community, and more broadly than that, the community that lives with these protists and these algae, and what they might be doing and contributing to those systems. So we can definitely offer quite a lot of expertise in terms of microbiome analysis, and also in biological interpretation of the organisms that you find in these cultures. We're also doing a lot of work with shotgun metagenomics, genome assembly. So as I say, I think we've probably got over 100 seaweeds and 100 cyanobacteria and microalgae data sitting on our servers at the moment. So we can do quite a lot to help there. When you do all of this sort of thing in terms, and I've mentioned phylogenetics and barcoding at all, but when you do this, you're interacting with a lot of, or using a lot of different bioinformatic tools. And so between Frederick, myself and other colleagues, we've been there and done that. So we can really help guide you in terms of what tools and potentially building bespoke tools or scripts to actually answer questions. But we're not experts. We are biologists and ecologists who do DNA sequencing and bioinformatics. We are not the experts. CCAP-ARIES is not a DNA sequencing facility. For that, we will definitely direct you to one of the NERC omics facilities. Typically for DNA and RNA, that will be Liverpool. And we will either direct you there or work with you and the sequencing facility and the bioinformaticians to achieve the results that you require. So that's kind of what we do now. But looking forward, we think there's a really major change coming, and that is big data. We've all heard this. In this case, I'm referring to the genomes of, or the blueprints of eukaryotes. We are eukaryotes, plants, animals, and algae and protists. It's over the last really decade, the ability to sequence large quantities of DNA and do it cheaply has changed hugely, such that we now have very ambitious projects being developed. This is the Earth Biogenome Project, conceived in 2018. And their aim is to sequence 2 million, the blueprints of 2 million eukaryotes from across the globe in the next decade. So it's roughly about 1,200 a week. And the UK arm of that Earth Biogenome Project is the Darwin Tree of Life Project. And CCAP is working very closely with the Darwin Tree of Life, and we're learning a huge amount, equally so are they. And we aim to submit, certainly in this first tranche, several hundred algal and protist cultures. And longer term, we hope to take that up to about 500. CCAP is working very closely with the Protist group there through the Earlham Institute, and we're helping in terms of DNA extraction and getting the quality of DNA that they require. But Fred and myself are also working with the Sanger assembly team to, I think, well, we're learning more than that, we're helping, but we are, every so often we can make more valuable contributions to that process. Because protists have proven to be, I think, if we had someone from the Sanger Institute, the hardest genomes to assemble. And I think, as Mikey's slide mentioned, where CCAP is also working with a number of other international genome sequencing consortiums. So we know that there's a lot of data coming, and it's only the beginning. And this is very exciting, but it also does represent a tsunami that it could overwhelm us. And the challenge is, how do we effectively interrogate and understand this data? And why we might bother and with the blueprints of protists, well, simply, while they might be invisible to the majority of us, they actually represent the majority of the biodiversity on this planet. So the things that we know about animals are about 4 o'clock on the in purple there, and then the plants are about 10:30 on in the green there. The rest of that cartoon tree represents the wheel of biodiversity on the planet. So protists really represent a massive resource for a lot of new discoveries, both in terms of, sort of evolution, in terms of ecology, but also in terms of sustainability and so on. A very strong theme that obviously we would advocate here at CCAP is a living culture is critical to being able to understand these blueprints. But this is echoed by far more eminent experts than ourselves, such as Patrick Keeling in UBC. These living cultures are necessary. We need to have them to be able to interpret and read off the blueprints of these protists and algae. I'm a bacteriologist. I can sequence the bacterial genome, I can read its code, and I can pretty much tell you what it's going to do and what it does. We can't do that for eukaryotes, the majority of eukaryotes. We've got whole departments devoted to just the human genome in Cambridge. We're talking about hundreds of very unique protists by comparison. So we need those living cultures to be able to modify their behavior and then look at the readout in terms of their genes and the metabolites, the proteins that they're making. And by that way, we learn how to read these organisms. Okay, so in the coming years, we're trying to build capacity in a number of areas. The first might be somewhat prosaic, but actually it's a big issue, is that a lot of this genomic data is actually very difficult to access, particularly if you're new to the field. It's spread across a lot of different places. So we envisaged that CCAP would be the landing point for you to then get your culture, and then we will identify where those resources are in terms of the genetics, and you can then go from there. Alongside of that, we hope to have some tutorials to really help you get started. And Frederick will talk a little bit more about this effort. So our first sort of gambit in this field, but this is something we want to develop. In terms of research, communities, systems is where it's happening. And there's a lot of interest in that, certainly from my own point of view, but from others. And so we want to get a lot better at being able to work with these communities. I'm projecting that in the next 5 to 10 years, pretty much every algal culture that comes into CCAP will either have a genome sequence or we will actually sequence it ourselves. So I believe that it's quite important that CCAP has the capacity to do that, because I don't think Liverpool is going to thank us sending our data to be analysed by them. I think it will quickly bog these facilities down. So do I have any time? No? Maybe, all right, okay. All right, so this is sort of kind of hot off the press. I think it's not a particularly good example of why having a living culture is important, but it kind of illustrates something that's happening right now that's kind of in my head. And this is data that hasn't been published by colleagues at the Tree of Life. So I'm grateful to Mark Baxter and Bethany for communicating this. So this is, so Bethany is analysing or trying to assemble the chromosomes or the genomes and the chromosomes of various eukaryotes, and she's currently working with this particular organism, Tetromyces. So this is now about to be published. So the genome note will be going out in the next few weeks, I gather. So the organism in the top right, there is a sort of an amoeboid thing. It probably won't show up in the light. And it eats bacteria. Nothing terribly unremarkable about that. But when you look at the genome, it is a diploid organism, so we're diploids, but actually it also has two triploid genomes. So it has a phenomenon called aneuploidy, which is uneven copy numbers of chromosomes. And that's not unique, but it's an unusual observation. And it might have some significance to somebody who's, for example, interested in cancer biology, because aneuploidy is a hallmark of cancer in us. And the point is, with this discovery, people can come and get the culture and actually sort of investigate the basis of that. Okay, I'm going to finish there. Thank you very much for your attention. Yeah, ask questions now or later in terms of what we can do and how we can help. Thank you very much.